Imagine developing a life-saving drug, one that patients depend on every single day. Now imagine that even a tiny structural change, something invisible to the naked eye, could affect its safety, effectiveness. So how do scientists ensure that every batch of a biopharmaceutical drug is exactly the same? In this video, we'll explore how peptide mapping plays a crucial role in making that possible. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, Canada. Welcome and Resolve Mass Laboratories we bring deep scientific expertise in advanced analytical characterization and complex molecular development to accelerate your IND and ANDA programs. Let's start with the basics. What are biopharmaceuticals? Biopharmaceuticals are complex therapeutic proteins produced inside living systems like specially engineered cells. These include monoclonal antibodies, insulin, and vaccines, some of the most advanced medicines we have today. But unlike small chemical drugs, these proteins are large and highly sensitive. Even small structural variations can impact their safety and effectiveness. That's why they require extremely rigorous structural verification. Now, the core problem, complexity. Here is where the real challenge begins. Proteins are not static. They undergo modifications like glycosylation, oxidation, and deamidation. These changes can alter how the drug behaves inside the body. At the same time, analyzing the intact protein often doesn't give enough detailed information. So scientists face a gap. They need a way to look deeper into the structure, and the solution: break the protein down into smaller, more manageable pieces. And that's where peptide mapping comes in. So think of it as creating a molecular fingerprint of a protein. First, protein is digested using an enzyme, usually trypsin or glucine or other enzymes, into smaller peptides. Then these peptides are separated using HPLC chromatography, and finally they are identified using mass spectrometry. The resulting pattern is compared to the reference profile. If everything matches, protein is confirmed to be correct. Now, how it works? Let's walk through this by step. First, digestion. Trypsin-like molecular scissors cutting the protein at specific sites. Next, separation. HPLC separates the peptides based on their chemical properties. And then comes identification. Mass spectrometry determines the mass and sequence of each peptide. And finally, matching. The data is compared to a known reference map. This entire process ensures the protein is exactly what it should be. Now, let's talk about essential roles in quality control. Peptide mapping plays several critical roles in quality control. It confirms the identity of the protein by verifying its amino acid sequence. It detects impurities or unexpected fragments, ensuring its purity. And most importantly, it ensures consistency, making sure every batch is the same as the last. This is especially important for regulatory approval and biosimilar development. Now let's talk about real world applications. In real world biopharma, peptide mapping supports high stakes decisions. When manufacturing processes change, like scaling up production, it helps prove comparability. It's also used to monitor post-translational modifications like oxidation and glycosylation. And in biosimilars, it provides strong evidence that a product is highly similar to the original drug, meeting strict regulatory requirements. Now, what are the challenges in mapping? Despite its power, peptide mapping isn't without challenges. Interpreting the data can be complex and requires expertise. Reproducibility can also be an issue if conditions are not tightly controlled. Even environmental factors like temperature or pH can influence results. That's why robust method development and validation are essential. Now let's talk about future trends. The future of peptide mapping is evolving rapidly. One major advancement is the multi-attribute method, that is MAM, which allows simultaneous monitoring or multiple quality attributes. We are also seeing the rise of AI-driven data analysis, automation, and high-resolution mass spectrometry. These innovations are moving peptide mapping toward real-time quality monitoring. So the bigger picture is, at the end of the day, peptide mapping is more than just a laboratory technique. It is a safeguard for human health. It ensures that every dose of a biopharmaceutical drug is safe, consistent, and effective. And behind every successful therapy, there is a deep level of molecular precision, making it all possible. For expert support in peptide characterization, impurity analysis, or regulatory-driven development, reach out to Resolve Mass Laboratory and let's advance your molecule together. Thank you so much for watching this informative video, and see you in the next one.